Hello everyone. Today we are going to learn how our body makes protein. It is very easy. Transcription and translation. First we have transcription. It happens inside the nucleus of the cell where the DNA is stored. Think of the DNA like a recipe book, but you can't take the whole book out of the library. So the nucleus, and so you make a copy of the recipe you needed, and this copy is the mRNA. It's like a little note with instruction. So the cell copies the recipe from the DNA, and the copied mRNA leaves the nucleus and goes to the ribosome and the cell's kitchen where the protein is made, and that is the first step, copying the instruction. Then translation, that is making the protein. Now how does it happens? This happens at the ribosome. The ribosome is like a chef reading the mRNA recipe to make a protein. Instead of using the sugar, the ribosome uses amino acid, the building block of the protein. Then how it works? The ribosome reads the mRNA recipe, and it helps the tRNA bringing it right amino acid. And the ribosome link to the amino acid together like beads on a string to make the protein. When the ribosome finishes reading the recipe, the protein is ready. So here's the big picture. Transcription is copying the DNA instruction in the mRNA, and translation is the ribosome reads the mRNA and build the protein with the amino acid. And that's how our body works. And it makes protein. And this protein help you grow, heal and stay healthy. So today we are going to know the molecular mechanism of protein synthesis, a vital process where genetic information encoded in the DNA is used to build protein. And this process occurs in the two major stages, transcription and translation. Let's break down. Transcription occurs in the nucleus, and it is involved in the synthesis of mRNA from the DNA template. This process is catalyzed by the RNA polymerase II in eukaryotic cell. Initiation happens. Transcription starts at the promoter region, commonly featured at the data box, about 25 to 30 base pair upstream of the transcription start site, and the transcription factor like TF2D, which binds to the data box, recruits RNA polymerase II to the promoter, and this forms the pre-initiation complex and unwind a section of the DNA, creating a transcription bubble. Then elongation takes place. RNA polymerase read the template strand in the 3' to 5' direction and synthesize a complementary RNA strand in the 5' to 3' direction. And the newly synthesized RNA use ribose sugar and substitute uracil for the thymine. As RNA polymerase progress, topoisomerase reveals supercoiling and RNA helicase unwind the DNA. Then termination takes place. Transcription ends with the RNA polymerase reach a terminal site signal, and in eukaryotic this is often involved in the polyadenylation signaling sequence AAUA-AAA, and this cleavage and polyadenylation specificity factor recognize the sequence, and the mRNA is cleaved downstream. Then post-transcription modifications takes place. In eukaryotic, the 5' capping, addition of the 7-methyl guanosine cap at the 5' end for the mRNA stability and ribosome recognition. Then polyadenylation takes place, adding of the poly A tail, that is the 200 adenine to the 3' end to prevent the degradation. Then splicing, that is the removing of the intron non-coding region and joining of the exon coding region by the spliceosome, a complex of SNRNPs, small nuclear ribonucleoproteins, and the mature mRNA is then exported through nuclear pore in the cytoplasm for translation. Then translation occurs in the cytoplasm at the ribosome, a complex made up of rRNA and protein. The ribosome have two subunits, large 60 and small 40 subunit in the eukaryotes. Then initiation takes place. Small ribosome subunit bind to the 5' UTR of the mRNA and scans for the start codon AUG. Initiation factor EIFS helps recruiting the first tRNA carrying methionine to the P site of the ribosome. The large ribosome subunit assemble, forming the complete ATS initiation complex. Then elongation takes place. The ribosome has 3 sites, A site, that is the amino cell where the tRNA carrying the amino acid enters. Then P side, the polypeptidil withholds the tRNA with growing the peptidyl chain. Then E side is the exit where it empty the tRNA. The ribosome read the mRNA codon by coding, and the tRNA bring the correct amino acid matching the codon via their anticodon. A peptide bond is formed between amino acid in P and A site, catalyzed by the peptidyl transferase. A function of 23S rRNA is in the large subunit. The ribosome translocates along the mRNA, shifting the tRNA from A to P and P to E side. When termination takes place, translation ends when a stop codon UAA and UAG or UGA is encountered. Releasing factor ERFs bind to the stop codon, triggering the release of the peptide chain. And the ribosome subunit disassemble and the mRNA can be degraded or translated again. Then what happens after translation? The polypeptide may undergo several modifications: folding, cleavage. In folding, it is assisted by chaperone protein to ensure the correct 3D structure, and in cleavage, removal of polypeptide. These peptides or a segment to activate the protein. Then chemical modification takes place. Phosphorylation, that is adding the phosphate group for activation, and glycoxylase, adding sugar for stability and recognition, and ubiquitination, that is the mark protein for degradation by the proteosomes. Then let's know the conclusion. The transcription, what does? It convert the DNA to mRNA in the nucleus, and translation convert mRNA to the polypeptide in the cytoplasm. The process is tightly regulated to ensure that the protein are made accurately and efficiently, and this mechanism are not just fundamental to cell biology but as a key to understand diseases and therapeutical development. The molecular mechanism of protein synthesis, it refers to the overall process of how protein are made from genetic information applicable to both prokaryotic and eukaryotic cell. It converts into transcription, that is converting the DNA into mRNA, and translation, that is converting mRNA to protein. It includes the role of enzyme ribosome, tRNA and regulatory factor without specifying the type of the ribosome. And now let's see the protein synthesis of 70S ribosome. It is specified to a particular prokaryotic and organelles. This focuses specifically on the bacteria, mitochondria and chloroplast, and thus use 70S ribosome for protein synthesis. Key feature of 70S ribosome, it is composed of 50S large and 30S small subunit. It is found in the prokaryotic cell, in mitochondria, chloroplast of the eukaryotic cell, and this process is fast and simple compared to the eukaryotic 80S ribosome translation. Mechanism of protein synthesis. It has been investigated most thoroughly in bacteria, usually in E. coli. Therefore, the process of protein synthesis of E. coli, which contains 70S ribosome in general described, and the protein synthesis of 70S ribosome in that, first what happened? Transcription. The process of protein synthesis started by the uncoiling of the strand of DNA molecule. One strand of the DNA molecule act as a template to forming the mRNA, and in the presence of RNA polymerase enzyme, the mRNA is formed according to the triplet codon of the DNA by copying or transcribing process. As soon as the mRNA is formed, it leaves the nucleus and reaches in the cytoplasm, where it attaches with the 30S subunit of the ribosome. The mRNA carries the triplet codon for the synthesis of protein. Then initiation of the protein synthesis takes place. Every mRNA contains at least one ribosome binding site, at which an active ribosome subunit assembled to establish an active ribosome. In prokaryote, it is the binding site, usually containing the base sequence AGGA, that is at the location roughly at 10 nucleotide from the beginning, that is the 5' end of the actual messenger. The 30S subunit bind first, and possibly with the help of the base pair between the 3' tail and 16S RNA, and the ribosome binding site in the presence of initiation factor, that is IF3 and possibly IF1. Initiation begins when the N-formyl methionine tRNA is accepted by the 30S mRNA at the AUG codon, which begins the messenger, and this step is promoted by the IF2 and GTP and possibly IF1. The 50S subunit is added last to the 30S mRNA Then F-met tRNA initiation complex, and this step results in the hydrolysis of the bound GTP to GTP and release of the initiation factor from 70S ribosome. The N-formyl methionine tRNA is a methionine charged tRNA that has been formulated at the free amino group of the methionine. Although there are two methionine transfer RNA, only one will permit the formylase enzyme that catalyzes formalization of its amino acid. Further, this F-met tRNA binds specifically to the normal methionine codon. The AUG, it will also bind less efficiently to GUG and valine codon. Although the AUG seems to be preferred, the starting single in the in vivo protein have been identified, whose message begins instead with the GUG. Thus, the same two codon that specifically orderly methionine and valine, when they occur in the interior of the polypeptide, they are responsible for the chain initiation. Then what happens? The transfer of amino acid into the site of the protein synthesis. Here what happened? The amino acid are transferred from the intercellular cytoplasm. This cytoplasmic amino acid pole, from there they are activated by the ribosome, by the transfer RNA molecule. So what happens? Amino acid are transferred from the intercellular cytoplasmic amino acid pole to active the ribosome. The ribosome are activated by the transfer RNA molecule. Before an amino acid can be incorporated into the growing polypeptide chain at the active ribosome, it must first be esterified to a suitable transfer RNA. That event is catalyzed by the enzyme. It is called as amino acid tRNA synthetase, which is specific for a amino acid in question and driving the reaction by coupling it to the hydrolysis of ATP to AMP. During the first reaction, the enzyme attached the amino acid to AMP to form the amino acid adenylate, that is AAAMP, by using a high energy anhydride bond between the carboxylate of the amino acid and the phosphate of AMP and releasing pyrophosphate. The amino acid adenylate complex is bounded to the enzyme until a second reaction replaces the anhydride bond with a lower energy ester link to the 3-terminal hydroxyl of the ribose of the adenosine at CCA end of tRNA. The amino acid, if we add with the ATP and enzyme, it will give amino acid adenylate enzyme plus pyrophosphate. Free pyrophosphate get hydrolyzed to PY by the enzyme pyrophosphatase, thus helping to drive the reaction to the right. AMP is released into the medium and the free enzyme can recycle to activate and attach another amino acid to another tRNA. In the free state, each amino acid tRNA synthesized preassemblly and has one site for recognition of its particular amino acid and one site for the binding with the ATP. After formation of the amino acid adenylate bond, the enzyme is believed to undergo a conformational change that unmasks another site at which the correct tRNA binds to the AAMP enzyme complex. DHU loop of the tRNA is found to have some role in the recognizing the specific synthesis enzyme, and thus for each amino acid there is at least one activating enzyme and at least one tRNA. That is, cell has 20 amino acid tRNA synthesis and 20 tRNA for 20 amino acid, and the charged tRNA or the amino acid tRNA move toward the site of the protein synthesis, that is the active ribosome. Then what happens? Polypeptide chain elongation happens. A amino acid tRNA or the charged tRNA is positioned at the mRNA by matching a 3 nucleotide complementary anticodon of the tRNA with the corresponding codon of the ribosome bound mRNA. In this way, the new amino acid is brought into a proper position with respect to the growing polypeptide chain. Only 2 sites of the ribosome are thought to accommodate tRNA at any one time, and therefore only 2 molecules of tRNA can be attached simultaneously to an active ribosome. The A site is the place of the entry where incoming amino acid tRNA is accepted at the ribosome, and the P site is the place of the release of the free tRNA, which is discharged from the ribosome after depositing it into the peptidyl chain onto a incoming amino acid tRNA. And at the A site, the tRNA is released after a sequence of action in which the peptidyl has been lengthened by one amino acid unit. Thus, during the chain elongation, codon-anticodon pairing bring a new amino acid tRNA into the A site of the ribosome, on which the mRNA rests, and this binding of incoming amino acid tRNA to the ribosome and mRNA involve hydrolysis of one molecule or of ATP like energy rich compound in presence of one non-ribosomal protein called the elongation factor, and T, that is the EFT. Actually, the two subunits called TU and TS, and the P site of the active ribosome is already occupied by the first coming amino acid tRNA, that is the F-met tRNA in E. coli. Now, peptidyl bond formation takes place to the incoming amino acid to the existing amino acid. Bond formation occurs by transferring of the first amino acid, that is FMT, to from its tRNA at the P site to the amino acid tRNA just brought to the A site, and this reaction is catalyzed by the peptidyl transferase. Once the peptidyl bond has been formed, the lengthened peptidyl tRNA is translocated back into the P site. Translocation requires hydrolysis of one GTP molecule and a translocase protein called as elongation factor G before peptidyl tRNA can return to the P site. However, now uncharged tRNA must be expelled from the place where it has been remained after discharging its peptidyl chain. Once this is accomplished, the ribosome restored to its previous state of readiness to accept an amino acid tRNA at A site, but the polypeptide is now one amino acid unit longer and is temporarily attached to the different tRNA that it was beginning of this process, and the ribosome moved to the next triplet codon after every cycle of peptide bond formation from the 5' towards the 3' end of the mRNA strand attached to the 30th subunit. The whole process of the chain elongation occurs every rapidly with the growth rate upon 40 amino acid per second in E. coli, and perhaps much faster in some eukaryotic cell. Example, 600 amino acid per minute in the reticulocytes during the hemoglobin synthesis, and the cellular rate of protein synthesis is enhanced even further by the presence of the multiple ribosomes on the same mRNA forming the polysomes. At this sequence of the events that takes place at the active ribosome during polypeptide chain elongation can be summarized as follows. Incoming amino acid tRNA enter the A site on the ribosome, recognizing the mRNA codon by its anticodon and then binding with it in the presence of one GTP or one elongation factor. Then peptidyl tRNA carries the nascent polypeptide chain occupying the P site. Then peptidyl bond synthesis takes place by transferring the polypeptide chain attached to the tRNA at the P site to the amino acid tRNA at the A site. Then formation of the peptide bond is completed, catalyzed by the enzyme peptidyl transferase, and the A site is now occupied by the peptidyl tRNA, while the free tRNA and the P site is released to the recycle. The peptidyl tRNA is translocated to the P site, and the process that requires GTP and elongation factor G. The A site is again free to accept the next amino acid tRNA. The ribosome is one codon further along the messenger, moving towards the 3' end of the mRNA, and the polypeptide chain is one amino acid longer after each cycle. Then termination and releasing of polypeptide chain takes place. That is, the termination of the polypeptide chain grow like its initiation takes place at a specific codon, that is UUA, UUG, UAG and UGA, and these are the nonsense codon that prematurely terminate translation when they appear by the mutation. Once a terminator codon is encountered, no further polypeptide chain synthesis occur. Recognition of the terminator codon comes not from the tRNA but from the protein called release factor, and one of which R1 is involved in the termination at the UAA and UAG, and the other, that is the R2 at UAA and UGA. When a ribosome encounters this codon in a presence of an appropriate releasing factor, the growing polypeptide chain is freed from the tRNA, and a reaction that required GTP. Hydrolysis of the GTP thus powered each ribosome associated step in the translation. The ribosome itself dissociates into an inactive subunit, an event that is mediated by the IF3 initiation factor. The dissociated ribosome subunit and tRNA are now free to participate in another round of polypeptide synthesis.